Okay, we'll get started. So it's good to have all of you here today, and this is a really fascinating panel. I think you will enjoy some of the topics that will be covered today. Really trying to see the transition, in terms of using these technologies, and how do you get them from either a thought process, just a thought process, to an actual action that you can take all the way to the patient. And we're going to be examining that using various strategies, looking at various strategies from our speakers. We have an absolutely superb panel today with us for this session. And the first speaker really needs no introduction. Dr. Randy Mills, has been in the field for many, many years. He basically was one of the first people to start RTI many, many years ago. And then, as you know, he went on to become the CEO at Osiris Therapeutics. And then currently he is actually the president of the California Institute for Regenerative Medicine. So I don't know of anyone really in the field that has done so many different things as Randy has, from researcher to scientist in a company to CEO and now in public service. So it's great to have Randy with us, Randy Mills, who's going to be talking to us about some of his experiences in this area. Randy, thank you so much for being with us today.
Let me know if y'all can't hear me. Yeah, okay? Okay, so we'll try to do it like this. So last Wednesday, I was in Washington, D.C. with my good friend Tony Atala, and as we were saying our goodbyes at the airport to go different directions, he says, hey, by the way, would you be on a panel with me next week? So that's why I'm here. This, what I'm about to show you actually has nothing to do with the California Institute for Regenerative Medicine. This is a specific request by Dr. Atala to tell you the story. And so if you don't like this story, it's not my fault. Blame Tony. Okay, with that, let's get started. So first things first, I am the president and CEO of the California Institute for Regenerative Medicine. CIRM is a $3 billion state initiative dedicated to the development and commercialization of stem cell therapies. It's a massive investment. There's no other single investment in cell therapies anywhere like it, not just in the United States, but throughout the world. We have 300 different technologies in various stages of development in our portfolio, for everything from discovery through multiple phase 3 pivotal trials. Very importantly about CIRM is we are a patient-driven and a patient-focused company. So our mission is to accelerate stem cell treatments to patients with unmet medical needs. We wake up every day and we try to think of creative and innovative ways of being successful on that mission. Now, with that out of the way, I'm going to tell you a story about the development of arguably the first commercial stem cell product to hit the market. And this goes back to my early days at Osiris Therapeutics. Now, to set the sort of the stage, when I came to Osiris, one, I was 32 years old, so I was pretty young. Two, I was the 7th CEO that Osiris had in the last 4 years. 3, the company was bankrupt. So we were kind of in a bit of a tight situation, and we had to think about things and do things differently than might otherwise take place. And so, sort of with that as the theme, when I got there, and Osiris was not a young company at this time. Osiris had already been around for 12 years. So it was a pretty well-established company. So about on my 3rd or 4th day, I get everyone in the company together in a room at Osiris, much like this, and I start asking them different questions about our development program. And one of the questions I ask is, so when is our first product coming to market? And the thing about Osiris is that it existed in those past 12 years largely as a research institution. I mean, it was almost indistinguishable from an academic institution in that the success was measured by the research studies done and the papers that were published, not by any development milestones towards commercializing products. And so when I asked this question, when are we going to commercialize our first stem cell product, I got absolute silence out of the group. And I kept pressing and eventually somebody raised their hand and said, well, maybe 2014. And I said, wrong. And they got mad. And they said, "You've only been here for 3 days. How do you know it's not 2014? And I said, "Because I've seen our balance sheet, and you won't have jobs if it's 2014. And it's sort of this reality set in. And so we had this conversation that day, which was Osiris University is closing, and Osiris, the company, is opening. And that did not sit well with a lot of people. And we went through, because we had to make this, after 12 years, you eventually, as a company, you do have to kind of get on with it. We were 12 years into this, and we weren't close to the market on anything. We had one product in phase 1. And so we sat and we went through these different sort of strategic planning and laying out models. And as we did that, some people made the turn nicely, and some people, as you can imagine, didn't make the turn. And those conversations would go pretty much the same way. They would come and say, "Hey, Randy, this really isn't for me. I'm not really sure I want to do this, so I'm going to leave." And okay, we're going to miss you. But we have to do what we have to do. Now, my belief, by the way, in commercialization, it centers solely around the fact that we had a drug at Osiris that was life-saving drug for a lethal condition called pediatric graft versus host disease. We actually brought the first stem cell drug to market, first approval anywhere in the world for that therapy. Median survival for that disease was 81 days. So the fact that it was taking us 12 years and we hadn't gotten anywhere didn't sit well with me, and I knew the only way that was going to be able to ultimately go on and help those children the way it should have was that if we started commercializing that product and commercializing it with urgency. And so as we were making these decisions on these different people were sort of opting out, or we were opting them out of Osiris, it became somewhat painful. Attrition is a painful thing to go through in a company. Now that was in July of 2004. In November of 2004, one evening I get this knock on my office door, and this young lady walks in. She had been out on maternity leave, so she had missed most of these things. And I wasn't that familiar with her. I knew she worked in orthopedics, and I knew she had an exceptional background academically. Rice undergrad, Duke PhD, Columbia postdoc, was an exceptionally well-trained scientist. And when she came and knocked on the door and she said, I can't do this anymore, I was waiting to hear the rest of the line about how she wants to go back into academia and do good research and publish papers. And I said, "Okay, yeah, so you don't want to do this anymore. What do you want to do?" She said, "I don't want to do any more of this just basic research. I want to commercialize something." And I thought I was being punked, because at that time, that was kind of the rage, And I was looking around for the camera. And I said, "Okay, well, what do you want to commercialize?" And she lays out this idea for this bone stem cell product. And it was a pretty believable idea. There were natural reservoirs of MSCs, and if you could isolate those MSCs and separate them out from other hematopoietic stem cells and contaminating elements and lipids, you could actually have a product that was a bone scaffold with a whole lot of healthy, viable MSCs on it. There's a lot of that to work out, but it was a reasonable assumption. So I listened to this story for about 10 minutes, and I said, "Okay, so you have another response." She was actually running what would be a whole nother clinical program. And I said, "Okay, so I'll let you do this, but you still have to do all of your other work. So you just work this in together and make it work." She says, "Okay, I'll do that." And she was leaving. I swear to God, every word of this is a true story. As she was leaving, I said, "Oh, by the way, when are you going to ship your first product?" And she turns around and she says, "Ship?" And I said, "Yeah, it's a revenue recognition thing. We can't recognize the revenue on any of this until it actually ships." And she does this. Now, keep in mind, the company had been around for 12 years, and we had gotten into phase 1. She says this, "Well, we couldn't ship it on a Friday because it's going to be a frozen product." Right? It's 12 years, she's worried about the day of the week we ship this product. So she does something in her head, and she goes over to this calendar, and she's fiddling with it, and she comes back, she says, "Wednesday, July 20th. It's November of 2004. She says, "Wednesday, July 20th, we're going to ship the world's first stem cell product." So I go to my whiteboard, and I have sort of things I want to keep track of and remember, and up on there I write, "Osteocell, Wednesday, July 20th, 2004, and she gets to work on developing this product. And this is what the osteocell looks like. It is a cancellous bone matrix that came from axial bone that was rich in MSCs. The per cc dose of MSCs had to be greater than 10 to the fifth. We had to show that, one, they were culture proliferative and that they would go down a trilineage assay. The average amount used in an application for spinal fusion was between 5 to 10 cc's, and it just happened to be the perfect environment for the adoption of this product, because my good friends at Medtronic Software Organic had just launched, or had previously launched, a product called Infuse, which originally was dominating in the market and then was running into a lot of safety concerns. So I write up on my whiteboard, Wednesday, July 20th, this young lady by herself in her spare time is going to launch something that this entire company for the previous 12 years couldn't figure out how to do. And we kind of chuckled about that until this. Wednesday, July 20th at 4:50, the first unit of Osteocell shipped out of Osiris Therapeutics, actually went to a surgery at the University of Southern California by an orthopedic surgeon there named Tom Vangs Ness. And the product went on to be a commercial superstar. So I never doubted her ever again. Anytime she said she wanted to do something, I've never seen such a great example of execution. So how well did it do? Well, this is quarterly how it did for us when we first launched it. We were basically backordered on this product from the word go. As I said, we had exactly the right environment for it. One company had a dominating BMP2. Everyone else wanted a biologic that was sophisticated. We had no idea at the time whether or not patients would want stem cells in them or avoid stem cells in them. It turns out, at least in orthopedics indications, they desperately wanted a stem cell based product in there. That product has to date gone on and treated over 250,000 patients. It has done 1.25 billion with a B in cumulative sales. It's still doing over $200 million a year in annual sales. It took us 8 months to develop, and I say us like I had something to do with it. It took us 8 months to develop and cost us actually less than $250,000 to develop. The product's, as I said, still on the market. We ended up selling the product after about 3 years of commercializing it. It brought into Osiris about $130 million net cash that we were able to then use that money to turn around and advance our research on developing our stem cell drug for pediatric graft versus host disease. So that's the Osteocell story that Tony wanted me to tell you. And the question I have, not is rhetorical, but to think about it. So how does that story make you feel? It makes some people very angry. It's sort of inspiring to other people, as I have clearly my own thoughts on it. But anyway, that's what I wanted to talk about. I was going to keep it brief, so I will. And thank you all very much.
Thank you so much. That was absolutely a great story, and you can see why I thought that would be a great setup for the session, because and to have Randy involved in this session, because it's just just shows how sometimes you just have to be very focused on what you do, and how this technology went all the way from a concept to delivery in a short period of time and was able to be delivered to the operating room. So our next speaker is actually Dr. Thomas Temple. I've known Tom for many years. He was at the University of Miami. He recently has moved over to a new position at Nova Southeastern University, where he's Senior Vice President for Translational Research. And Tom was really instrumental in the development of the tissue bank at the University of Miami, which then actually was bought out as a commercial entity, and is now a very, very important tissue bank in the national scene. So, Tom, thank you so much for being with us today.
So, allografts have been around for a very long time, and what you see is a 5th century depiction of Damien and Cosmos, the surgeon and physician twins who allegedly had performed this first homograft transplantation in a church sexton. Since then, the field of allogeneic tissue transfer has exploded and has become one of the mainstays in orthopedic oncologic reconstruction. I'm just going to share a case with you to show you how this field has advanced and the kinds of things that have made it possible. This is a 2-year-old child with a Ewing sarcoma, a highly lethal form of bone cancer. And here you can see his preoperative image on the upper left. And you can see that the entire femur is involved from the hip to the knee. The PET scan is illustrating significant avidity throughout the left femur. And really, there are not many limb salvage options in a child affected with this condition. So what we opted to do is utilize both anatomic and physiologic imaging to assess the efficacy of advanced chemotherapeutic intervention. So what we thought we would do is see if we can get a very good response and allow this child to grow a little bit. What you could see here is that, in fact, he has grown from the previous image to a later image. So we treated him with about almost a year of chemotherapy. And you could see that he had very little avidity in this area. Again, a very extensive tumor, physiologically excellent response to chemotherapy, and allowed him a period of growth to allow us some bone to work with for the reconstructive purposes. So this is just an example of how we're able to resect this entire femoral diaphysis. Here you can see the very close margins, distally as well as proximally. And then a reconstruction. This happened to be a femur from a 3-year-old child who had died several months before in a drowning accident. So here's the initial reconstruction, and then over time we've been able to preserve the growth plates and allow this graft to grow along with the child. He's now 7 years out and continues to grow and has a ostensibly normal life. So this is a great advantage of tissue transplantation, the use of a reconstructive and regenerative technology in the operating theater. But they're not always so wonderful. Anybody who does this for a living knows that fractures occur in up to 20% of patients with structural allogeneic tissue transfer, and infections occur with alarming frequency as well. So it sort of moved us as a bank and as a field into the regenerative technologies, where we're able to use injectable osteoinductive and conductive materials for bone defects and develop new implants and structural orthobiologics for reconstruction, and then finally to engineer natural and synthetic matrices along with stem cell platforms to achieve the same purpose. One of the things that we developed was the microparticulate bone. And the question that we had was, does size matter in the scheme of bone development? And what we found was in a study using non-human primates, using the distal femur from these baboons, so we created a defect, a closed segment defect, and then we filled it with various particle sizes from between 100 and 300, less than 100, or 5 to 800, 800, 1200, and actually looked histopathologically at a focal point in time as to the amount of mesenchymal cell recruitment, new bone formation, consolidation. And what we found from this study was that, in fact, a composite of between 100 and 300 microns was optimal in terms of mesenchymal cell recruitment and new bone formation. And here you can see on this nomogram the 1 to 300 micron configuration compared with the gold standard at the time, autogenous bone graft. And then in the next 1,000 cases, we utilized this matrix, and this is just an example of a woman who had a large aneurysmal bone cyst that was resected. This is just the graft at time 0, and this is the graft at 6 weeks. And you could see that the regenerative capacity of this matrix, so long as it was either juxtaposed or within a bone cavity, was pretty significant. Another technology that had been floating around the University of Miami for many, many years, to the previous speaker's point, and had gone nowhere, was this Miami cell, this marrow-isolated adult multilineage inducible stem cell that has these markers that make it common to stem cells and unique to this particular cell. And this cell, it can cross germ layers and form bone, cartilage, neurons, and even hepatocytes and skin. What's also interesting about the cell is that when you look at it on a microarray and compare it with bone marrow cells as well as embryonal stem cells, you can see that many of the genes expressed in this Miami cell are also seen in embryonal cells. The difference between this and embryonal cells, it follows a script. And the other interesting and necessary facet of this cell is that it does not express MHC1 or 2 antigens. So again, two technologies at the same university working in different silos come together, but commercialization would have been very, very difficult had it not been for the company Vivex, who actually recognized the value in a matrix and a stem cell, put it together in 18 months, developed a commercial stem cell product. And this is just some of the preliminary work in perfusion cultures demonstrating these small microparticles with the Miami cell. And here you can see that curiously they resorb just a small fraction of these sharp edges of the particles, the cells, and they form these bridges, these intercellular bridges between particles that eventually coalesce, as you can see in culture. So the question is, could we take this Miami cell and this microparticulate bone and create a product? And we did, one called Viagraft. And these are basically viable cells that support osteogenic healing process. They have extraordinary osteogenic potential, and they work well within a framework of a three-dimensional osteoconductive matrix. And this is just an example of the frozen cells mixed with the micronized bone, the 1 to 300 particle sizes, and then placed, in this case, into an allograft host junction non-union. And what makes these cells so robust, we think, is the very large percentage of bone-forming mesenchymal cells with every aliquot, as well as the Miami cell, which represents a relatively small portion of it. Just as an example of how we can use these stem cell technologies in the operating room, this is a 38-year-old male who sustained a pathologic fracture through a low-grade osteosarcoma while skiing in Vail. Here you can see the pathologic fracture through this lesion in the tibia. Here's the resected tibia, the bivalve tumor. Here you can see the reconstruction. This is actually a structural allograft with a vascularized fibula, and what's strikingly inconspicuous in this is the absence of adequate fixation into the graft. So this went on predictably to form a non-union at the proximal allograft host junction site. And here you can see the preoperative image, a large 5 millimeter gap, and here immediately post-op. Here at 6 weeks, you start to see some extraperiosteal new bone formation, which is a little bit better here. Pre-op, 6 weeks, extraperiosteal new bone formation, and again, a higher power view of that. Then at 3 months, you can start to see consolidation across the fracture site. And then at 6 months, you see complete consolidation. And this is something that would have been very difficult to do even with autogenous bone grafts. So this is an extreme example of a significant problem that's very difficult to affect healing down the road that we were able to achieve with this allogeneic stem cell composite. This is just another example of a patient with a myeloma, and here you could see this has been irradiated and the bone is very thin, so there's an impending pathologic fracture. You can also see that the glenohumeral joint is subluxed substantially, and this patient was on the operating schedule for a proximal humeral replacement, which would involve removing the entire upper portion of the humerus and reconstructing that with a metal implant. But instead, through a 1 centimeter incision, we were able to apply this composite matrix with a stem cell. And here you can see 2 weeks post-op, 10 weeks post-op, and what you see now is that the glenohumeral joint is now in a normal location because the muscles are developing around the joint, and then at 1 year post-op, ostensibly normal function. So again, a very difficult environment for bone to form, radiated bed, pseudo-subluxation, impending pathologic fracture, but able to affect healing with a very minimal surgical intervention. So as we look at future iterations, we can envision synthetic and biologic composites that are composed of ceramics and bioglasses and allograft to form these multifunctional materials that combined with stem cell products not only form new bone but supporting structures as well, like the vascular neurospace, to create a perfect reconstructive option for patients in need. Thanks very much.
Thank you, Tom, for that presentation, and very, very informative. And our next speaker is Dr. Boulis, and he basically did his undergrad at Harvard, then did some training at Michigan, and now is practicing medicine and surgery. And his area of expertise has really been in the neural regeneration space. And I think you will find the work that he's doing fascinating. Thank you for being with us.
Thanks. So I'll come back to the question was posed by the first speaker. How does this make you feel? First of all, impressed. I love those kinds of stories, but also deeply bitter. You know, as a neurosurgeon, I wake up every day, somebody with an interest in repairing the nervous system, and look in the mirror and ask myself why I couldn't be interested in bone. The portion of neurosurgery that operates on the spine tends to make a lot more money than those of us muddling around in the nervous system. In any case, you know, this repairing the nervous system thing is an enormous challenge. It's in fact, you know, when I was growing up, we were told that the nervous system didn't really make new cells, and what you got was what you had, and if you damaged it, you're out of luck. So the stem cell revolution has really been about challenging that. Personally, spent most of my time actually in trying to optimize around the cells at hand, to try and solve some of the problems that we face as we try to develop a clinically practical application for stem cells, in particular in the spinal cord. And so I'm really going to focus on the story of the device development and how we optimize the cells, but I'll take a brief moment to introduce the main cast of characters that has entered the world predominantly of ALS, stem cell transplantation, the spinal cord, which is what I've worked on. But just to make the point, I think the spinal cord really has a collection of pathology that we can go after with cell-based technologies. Spinal cord injury, where we're going after trauma, trying to repair white matter. MS, where we're dealing with an autoimmune problem and we're trying to replace oligodendroglia. And then motor neuron disease, which is a neurodegenerative process in which we lose motor neurons, and in fact, the defect may be in the glia, and so glia may be a target for replacement in terms of neuroprotection. In the long run, to rebuild this problem, we're going to have to replace motor neurons. We understand a good deal about the mechanisms of ALS. We know that there's excitotoxicity at play, where there's too many excitatory amino acids, and that may lead to the death of cells. We know this because the only drug that works in ALS is a drug that works against excitotoxicity. That may be because of impaired glutamate reuptake on the part of the astrocytes, which is one of the reasons why replacing astrocytes may be a good idea in this disease. There are also toxins that play a role, both environmental toxins, as we've been able to see that there are certain populations that seem to get this disease in greater numbers. For example, during Gulf War, there's a substantially increased incidence of ALS, suggesting that environmental toxins can play a role, as well as secreted toxins. Microglia, or the immune system, seems to be important. That's where the term amyotrophic lateral sclerosis comes from. Clearly, adjusting the immune system may play a role as well. And then finally, the genetics of this are being increasingly well understood. There are a variety of genes that have contributed to familial ALS that have been identified, and over time, we've begun to recognize that there are some genes that may play a role in sporadic ALS as well. As you launch into trying to design surgery for the spinal cord, to do repair, you have to buy into the idea of segmental therapy. And what I mean by that is ALS is a disorder of both upper motor neurons and lower motor neurons throughout the spinal cord. We're not going to be able to do surgery on the whole neural axis. So this requires that you buy into the idea that patients are dying because they're losing the ability to breathe and control their upper airway, and that keeping them, and that can be affected by transplanting cells into the C3 through 5 region. If you want to preserve upper extremity function, you got to go C3 through perhaps T1, but that's a local area, local segments, and that's probably the most important point. But also, if you want to keep people walking, you got to go for the lumbar enlargement. So you can choose segments within the spinal cord to go after. We have these animals, SOD1 rats and mice, that model the disease extremely well and allow us to test these approaches. I just threw this slide in there to make the point that, of course, we can try to replace neurons, we can try to replace astrocytes or oligodendroglia, which are the myelinating cells. All of them seem to play a role in ALS, and each individually may be important, and some of these cells will differentiate into to a certain percentage of all of these. And that's one of the advantages of stem cells. It's not, I mean, they can do a lot of different things, which this kind of cocktail approach may be what's called for. So what about motor neuron replacement? What about these patients who are already advanced in the disease? What is their hope? Well, that we could potentially rebuild the nervous system from the spinal cord out. And I would submit to you that that's the simplest place to start in the nervous system, because really what those cells are doing is getting a descending message and sending it out to muscle. It's not a complex computational structure. So in theory, this ought to be the easiest place to rebuild the nervous system. And we do know that you can make motor neurons, starting out with embryonic stem cells or other forms of stem cells, using certain signals to drive motor neuron production. This is the work of Doug Kerr, in which he started with GFP animals and stem cells from them, so that you could see what happened with those cells. And here you're seeing axons, projecting out into the periphery and forming new neuromuscular junctions. What was really impressive in that work was it's not going to let me do it, is it? Is the recovery of function in those animals. What you're seeing here is the side transplanted. You'll have to take my word for it. The side transplanted, the animals began to move their limbs, and you could see those green fluorescent axons going out into the periphery, forming new neuromuscular junctions. So it is possible, but this is going to be an extremely high hurdle to get over. So most of the field right now is really focused on trying to protect the existing motor neurons to prevent the progression of disease. That's where we focused. This comes from early work from Clive Svensson's group, in which he's using neural progenitors which are taken from human cortex or brain, and using lentivirus to induce the production of glial-derived neurotrophic factor. And he's able to show, so there you can see the expression of GDNF from this graft that he injected here, and what you can see is that you see preservation of motor neurons on the side where GDNF is being made, but not the other side. Turns out that these cells are not terribly effective at protection when they're not producing GDNF. These are the cells from NeuralStem, headed up by my friend Carl Joes in the audience today. So this is some of the original work showing the improvement in the survival curve of these SOD1 animals after transplantation and preservation of motor neurons after transplantation. That was the preclinical proof of principle data that led to the NeuralStem trials, where we've gone as far as phase 2, and I'll quickly go through that. There's this other group working with, God, I hate it when that happens, working with glial-restricted precursors. And this is the strategy of Q Therapeutics. Here again, you can see the shift in the survival curve of these animals. These cells have not only gotten FDA approval to go into the clinic, but have also recently gotten funding. So these guys are going to be going into the spinal cord probably in almost certainly in 2016. So as we begin to ponder how do we do these transplants, that posed unique challenges. Let's see if I can actually. Sorry. Okay. Well, let me make the point here. So this is an American that went to Beijing to undergo transplantation of oligodendroglial or olfactory and sheathing cells. So it's a proto stem cell type cell. The way that the surgeon was going about doing this was really just to take a butterfly needle, stick it in Eppendorf, and then suck back on the cells, and then take that with a hemostat and place it into the spinal cord and inject with a tuberculin syringe. So, kind of having reviewed that, we tried to decide basic principles that would improve matters. Now, the problem is thickness of the spinal cord is only 8 millimeters. So if you go in the 4 to 6 millimeters necessary to be in the region of the ventral horn, and you inject a volume, help in a handheld, with a handheld syringe, most of that payload is going to come right back up the needle and dump into the subarachnoid space. It's not going to go where you want. Your targeting is going to be extremely inconsistent. So we developed this idea that in order to keep the cells in the spinal cord, you needed to pump the cells in very slowly, number one. That if you're going to pump in slowly in the spinal cord, that you need a fixed platform that will move with the patient, because if you're going to try and hold that in the spinal cord over a long period of time, you're likely to cause shearing, which will paralyze the patient, and hence you're going to be worse off than when you started. So we developed a fixed platform, and that this same fixed platform would allow for reproducible targeting, which is going to be necessary for this to work in the long run if we brought it out to many, many surgeons. The methodology was to go 2 millimeters, 1 to 2 millimeters medial to the identifiable dorsal root entry zone, drop in about 4 to 6 millimeters, and then your cells will be in the ventral horn. So we went on to design this. This is the second platform that we designed. We were able to show that we were able to hit our target fairly consistently, and that animals, this is a behavioral scale. While there's some deficits early on, that all the animals came back to baseline. As we talked about this, we optimized this platform by using percutaneous posts to mount to bone. That allowed us to reduce the incision size, and by elevating the platform up out of the incision, we were able to make it wider, so we had better visualization to go in between these rods and work on the spinal cord under a microscope, and using this sort of ball turret to get that orthogonal trajectory that's necessary. So we designed this platform, which is our third platform, and here you can see it on a pig being deployed with the Z drive here dropping the needles in. We then designed this novel cannula, which we call the floating cannula, which really works like a harpoon, which is to say that it goes in rigidly, and then you can slide back the outer sheath, and then it's flexible. Okay, and that was necessary because it turns out that while this platform moved with the patient, so you didn't have to worry about shearing, the spinal cord is actually moving in the canal as patients are breathing and as their heart's beating, so that would shear as well. So the floating cannula took advantage of this. It also added an extra level of safety, because if for some reason that system got bumped very strongly, it would bend and the thing would pull out rather than shearing across the spinal cord. Lastly, we created this flange that retarded reflux to try and keep the cells in place. We went on to test this again, animals all recovering back to baseline, and our targeting, as you can see here, was fairly consistent and accurate. So having this methodology and these devices, we felt we could move on into the clinic. We've explored how far you can go in terms of volume, and surprisingly we've learned that volume doesn't really seem to have a dose-limiting toxicity. And that's probably because as you get to a certain volume, you experience this reflux. You don't actually keep the cells in place. But if you go and you look for the optimal volume to inject, it turns out 25 microliters is probably where you peak out in terms of the best delivery with the least damage. We've studied that. Again, we've also looked at this, and we expected that as we escalated numbers of injections that we'd also hit dose-limiting toxicity. That turned out not to be true too. If you have a good method to go into the spinal cord, then chances are you can repeatedly do it. So this comes back to the segmental therapy idea. It turned out that we were able to do far more injections than we initially expected. And that's a story of development too. In my mind, when we first launched into this in humans, I thought we were going to do a single injection and see whether that was safe. And it was the guys at Neuralstem that said, that is not ever going to be a product, so you need to get beyond that, and sort of challenged us to get into this territory where you do many, many injections. It turns out, as we've studied this, that accuracy really matters in this. That if you hit gray matter, the cells spread centripetally. But if you hit white matter, they spread out and run along the white matter tracks as if they were almost a railroad. And we know that that's sort of an active process. It's not just passive spread of the cells. For example, when you see here, cell that the injection just hits the edge of gray matter, the cells almost sump out into gray matter and migrate along in that direction. So if you're trying to repair white matter, that's great. You got to hit white matter. so you can get the cells to spread out. If your cells are going to be secreting something, you might want to hit white matter so that the white matter forms a means of distributing those cells. But if you're going to replace motor neurons, you better hit the gray matter. Or if you're going to protect the motor neurons with your cells. So this becomes very important. So we're able to move through both large and small animal preclinical tox studies, and that got us into our phase 1 trial, going into the lumbar cord. The principles of this were risk escalation. While normally with a drug you just do dose escalation, in this case operating on the spinal cord has all these different axes of risk. So you're best off starting low because you can't make somebody quadriplegic. You're best off starting unilateral because it's safer. And you're best off starting in non-ambulatory patients because you can't put them in a wheelchair because they're already there. So we really moved gradually through these various groups until we were finally allowed to do injections of the cervical spinal cord where the risk was quadriplegia. And we were able to show that we could do that safely. And this is a diagram showing how we escalated initially in non-ambulatory patients unilateral, then bilateral, then going into ambulatory patients unilaterally, then bilaterally, then moving into the cervical spinal cord, and finally doing a staged lumbar cervical injection, which is our therapy, which was to preserve breathing, preserve proximal arm function, and then also preserve walking. Immunosuppression, we used a huge, very powerful cocktail recommended to us by our whole organ transplant colleagues, but we're pretty certain at this point that you really don't need that much, and there's a variety of evidence that supports that. This is the first patient to be injected. We use somatosensory evoked potentials and motor evoked potentials in the operating room to monitor the spinal cord as we're doing this, and we have certain rules for stopping. Knock on wood, we've never really had to do that. This is the assembly of the platform in a patient, the use of fluoroscopy to localize where we're going. The use of the floating cannula that actually backloads cells using a pump that has a built-in vibrator to keep the cells in suspension. Again, as we go through this, the story is, well, the cells are great, but there's all these problems. How do you keep them in suspension in your device that's doing the injection so that you get homogeneous delivery? And then how do you make sure you're accurate? And then how do you know where the cells are when you're done? These are the first 5 injections in a human. As we move forward, this was the device as deployed on the cervical spine. As we finally got there, patients arranged for that. I'm not sure if this is going to work given how miserably the last one worked. Oh, actually it will. So you can see the spinal cord here moving, and here's the needle going into the spinal cord in slow motion, obviously aiming between the vascular structures, and then pulling back the outer sheath so we have that flexible cannula in place. And then because of the fixed platform, that when the patient moves, you can see that the needle's moving with it, so that prevents shearing across the spinal cord. The other point with regard to the floating cannula that I made earlier, hopefully you'll be able to see here, that as the patient's breathing, you see a shift in the spinal cord in the canal, and you can see how this flexible cannula actually allows for movement with the spinal cord in addition to helping retard reflux. So this got us into our clinical trial, which we've published. Those results allowed us to do a more typical dose escalation, which involved really increasing initially the number of injections and then the concentrations. Everybody wants to know how the trial worked and whether that caused efficacy. So what we've actually done here, even though the numbers it's far too underpowered to really make those conclusions, is to compare to historic controls from much larger pharmacological trials. And I'm just going to show you this data where we're looking at the slope of the ALS rating scale in comparison with these historical controls from much larger groups, and asking how many of the patients were above what you would expect or did better than expected versus worse than expected. You see in our phase 1-2, oh God, it's miserable, in comparison with the ceftriaxone trial, that it was about the same. But if you looked at the PROACT trial, our patients did substantially better. And when you looked at Grift strength, as opposed to the ALS-FRS, comparing to the ceftriaxone trial, we did substantially better. The other thing is that it turns out that the forced vital capacity or breathing seemed to correlate with the ALS-FRS. So we do think that there's been a whiff of efficacy in this, enough to justify moving on to a phase 3 trial, and that's what we're planning now. I guess the last things I really want to show you with regard to this issue of, continually improving the methodology of delivery to get better and better homogeneity of targeting and making it easier for the surgeon. This is the new Gamma platform, the fourth version, which is now going to have little wheels that allow you to adjust in your targeting, and it's just a much more user-friendly device. It's going to allow for locking onto the Mayfield head holder using the Buddy Halo adapter system, so that we won't have to screw into the back of the head anymore. We think that'll get more rigidity. This system for the Z drive will also allow us to quickly switch out, so we can load in more cells as we go, and will also allow us to do potentially double injections if possible. Last thing I want to show you is that actually identifying these cells or tracking these cells has been a real challenge. Now, one of the advantages of working on a terminal disease like ALS is when the patients die, we can recover their spinal cords to see what happened to these cells. So it turns out that you can take video from the operating room and know exactly where each injection was done based on the fingerprint of the vasculature on the surface of the spinal cord, so that when you go back in the post, you know exactly where the injections were done, and you can go and actually hunt for the cells. So we have actually been able to see these collections of cells. The question is, are those graft cells or are those some kind of autologous cells? And the answer is, in this case, where it was a female that died, we were actually able to stain with the Y chromosome. We know the cells are surviving out several years. You can do FISH analysis. We've also done DNA fingerprinting type approach of looking for donor-specific alleles to look for the amount of DNA that's there. So we know that these cells are surviving. But this is still a problem. So we've begun to study how might you label these in a way that you could get into the clinic extremely quickly. We're using magnetic nanoparticles, optimizing the sequences to do that, studying where they go. And in the end, I'm going to show you a picture. Now where we're doing high dose of the nanoparticles, same amount of cells versus low dose versus none, you can actually see exactly where your injections are. And that, to me as a surgeon, gives me a lot of comfort that I actually hit that target. Remember that targeting is very important. So I guess I'm going to leave it at that. Oh, no, I do want to show you one last thing in terms of optimization, which is that we've begun to be able to do IMRI-guided injections, which is to say that we can actually use an MRI scanner using this non-ferromagnetic platform to begin to target the spinal cord. Here, that's a phantom that we're injecting. You can see the nanoparticles going in, hitting the target, and we've begun to do that on large animals. And you can see here cannula going in the MRI scanner. Here's the cells being infused. Now the needle's removed and you can see the cells hit the target. We're able to do this without actually opening a skin incision. So hopefully in the future, I think this is going to be really great, because if you're, for example, trying to remyelinate or go into a MS plaque, you can't see that from the surface of the spinal cord. So you as a surgeon expose that cord, you still don't really know where to go, but the MRI can see it very well. So this may be a really nice indication for MS, for example. It also may be good for re-injection, where if it turns out that the cells cause a repeated effect, going back in through a prior laminectomy may be a lot easier with an IMRI type of technique. So that's our story. We're through phase 2, trying to get into phase 3. Q Therapeutics is getting into the clinic soon. Dr. Svensson's project at Cedars-Sinai in LA is going to be in the clinic soon too as well. So the story will continue to evolve.
Thank you, Nick. Thanks for that nice presentation and nice work. And seeing the translation of these technologies all the way to the operating room. That's really great. So we'll ask our panelists to come up, and we're going to do two things. The first is to go ahead and open up the floor for questions from the audience, and then we will enter into a panel discussion. So first we'll go ahead for a Q&A. Yes, please, come right ahead.
Actually, Dr. Joe's here too. He may want to comment on this, but I think, the FDA seems to have been predominantly interested in the toxicity issue. One, are the cells forming tumors? Two, are there karyotypic changes in the cells over time? that might suggest that the cells would do something unpredictable. 3, what about the surgery? You know, are we seeing injuries in the process? So they're really focused on that. They didn't. I don't recall them ever being terribly concerned about, can you identify these cells post-mortem. On the other hand, I think, you know, some of our funding agencies, as we move through this, have been particularly interested in this, because I think they recognize that something like neural repair is sort of a long road, and there may be missteps, and actually being able to see, did the cells survive? What did they turn into? Where did they go? All of that stuff is going to be really important in the humans. But you know, of course, the FDA is interested in that. But we can track human to rodent transplants or human to pig transplants. It's a frustrating thing, because those graphs don't necessarily model what's actually happening as in the allograft situation. So we have done pig to pig also. We went after nanoparticles, iron oxide nanoparticles, because ferriheme is actually FDA approved as a way to supplement iron. So we felt that the pathway to bringing these two things together and getting into the clinic would be the shortest. And we knew we wanted to have confirmation, was the graft getting in as we pump, and were we hitting our target? And then we wanted some idea of whether it was sticking around. And then post-mortem, we wanted to be able to say, okay, those are the cells. Oh, there's the nanoparticle. Oh, look, it's in a macrophage now. The cell must be dead. So that's where we're trying to go with it. They tend to they tend in gray matter to move about a millimeter in either direction, and that's the kind of thing that, you know, as a surgeon, you hear that, and that brackets your idea of how many injections you need to inject. I have to say, I earlier in this process, I thought, well, you cell guys are going to develop a better cell that migrates over a bigger distance, and that'll be great because I won't have to do as many injections. And then suddenly, like an IMRI technique is going to be a lot better because we can go in once and inject a lot and have the cells distribute. But then, as we've studied this, we realized that reflux is not our friend, and in fact, you can go to higher volumes, but you're not actually going to deliver a whole lot more cells. So I've kind of shifted over time to, you probably do have to do a whole lot of injections to keep the cells in place, unless somebody comes up with a better way of doing that.
Thank you. Yes, one more question. Please go right ahead.
The cells are allogeneic, marrow-derived. They're not expanded. We basically isolate them from the marrow, and then we put them through a Ficol gradient. We take the stem cell layer out, and then we put that into a low O2 environment in a selective media. We don't expand them. And then they go back into the same microparticulate from the same donor. So they're homologous, non-expanded. And in the last 15 years, all the work that we've done, we've never been able to demonstrate tumorigenicity with these cells. And I've used them in patients who've had radiated beds, patients who've had chemotherapy, wide excision, biologic reconstruction, and we've seen no adverse effects.
Okay, so we're running out of time, so we're just going to go ahead and try to ask one question of all the panelists, and Randy, I guess I'll start with you, and the same question basically. What do you given today's environment in regenerative medicine, given all the history we've had before us, and what we have in front of us, what do you think are some of the things that we should be doing to have more success stories like the ones we've heard in this session?
Well, I think the thing that concerns me, we were talking about this last week, is that the therapies that we see currently in development in regenerative medicine represent only a fraction of those therapies that potentially could be involved if more people were getting involved and more people were committing resources from a corporate level to the development of regenerative medicine. And I think one of the reasons that we don't see that is because so far the regulatory paradigm that exists, we have to just call it for what it is. I mean, the scoreboard is very clear. It's been in place for 15 years, and there is a 0 up on that scoreboard for stem cell therapies in the United States. And when a regulatory paradigm is viewed as prohibitive, not just from I'm not suggesting it's not possible to accomplish, but I can tell you at Osiris there were numerous occasions where we never attempted to develop technology we had very strong beliefs would be effective, because of the time and the financial commitment that would be required would never be able to be made up, because the patient populations that regenerative medicine tends to go after tend to be pretty small. And so I think that is one aspect that we need to have a conversation about honestly, and figure out what we can do, because 0 isn't okay when it's a life-threatening disease.
Thanks for those comments. Randy, we'll go to Tom next. Tom, what would you say? What would you say would be the things that we need to be doing to get some of these technologies further into patients?
Yeah, I would agree that we need a better regulatory environment to basically harness some of these ideas and sort of move the needle forward. I think that we've really had to dumb down some of the technologies to make them appropriate for the regulatory auditors, and I think that the INDs are so prohibitively expensive that it really does make it very difficult. So I think those are the kinds of things that we need to think about. I think that the regulatory issues really are the major stumbling block in moving forward right now.
Thank you, Tom. Nick?
So I actually gave a TED Med talk which focused on the issue of regulation as a barrier to the development of personalized medicine and orphan disease type of structures. So I'm on board with these guys. The other thing I would say is, take the CIRM concept national. I mean, not everybody can live in California. I mean, I think it was a brilliant idea on the part of the people of California to seed the development of that industry. It's been almost impossible not to move to California. But on the other hand, I think the availability of funding that moves, that actually joins the late-stage translational process to the early-stage clinical trial process is at least as far as I know, revolutionary. I mean, I look at the at least extramurally funded route for developing ideas and coupling preclinical development IND application or IND acquisition to your phase 1 trials is a brilliant move, because why stop once you have your approval? You should just keep going, not just like, okay, now I've got my IND, now I'm going to apply for funding again, and that adds another 2 years of iterative processes. So
Great, thanks. Maybe we can ask audience participation on this panel, because I think this is an important topic. How many people in the audience would say that the major challenge, and we're going to give a multiple choice, with the multiple choices for the major challenge, what do you think is a major challenge in the field for the future? Is it cost of the technology, or is it the regulatory challenge of the technology? So how many people think it's the cost of the technology? So raise your hands, please, and keep them up, and you can look around. And okay, and how many of you believe it's the regulatory aspect of the technology? Wow. Okay, that's overwhelming. Well, I think there's a lot of,
Can I, Tony?
Yes,
I just want to point out that those issues are linked, right? I mean, the cost of a therapy being developed for a period, we don't know how long it is because it's not been done. We know it's more than 15 years. It makes it, so just the cost of carrying that amount of capital, if you believe the $2.6 billion number, is about $250 million a year in just time value of money. So there is no chance. This is important to understand. There is no chance at bending the drug cost curve without changing the regulatory paradigm. People are going to be, I don't believe how much this drug costs. Keep believing it until the time to develop a drug gets shorter.
And
I also think it shifts a little bit as you move from, like, when you're looking at phase 1 trials, the big hurdle is regulation, which is where most of us are. When you're looking at phase 3 trials, now you're facing a really big financial challenge. I mean, trying to fund at least a surgical stem cell trial for hundreds of patients is extraordinarily expensive. So, you know.
And it's linked, right? Because if you look at the pharma industry, right, and you look at the numbers, if you believe the numbers from the latest study from Tufts, the average time to develop a technology, a pharma, just a drug, a pill, is about $1.1 billion over a 15-year period. So yes, that's actually, if you include all the. So basically, the interesting thing is that, we are in the same, we're looking at the same challenge for RegenMed as well, and even higher challenges because of the regulatory path. So I think it's been a great discussion, and I guess, as Nick said, we can't all be in California, but we can all come visit you, Randy. So it's great having you all on the panel. Thank you so much for all the participants, and thank you for your attention.